This is the video for the Micro Lab Special Disease Project, and my topic I chose is the plague, or the Black Death as it's commonly known as. So I want to start off with a little bit of history. When people hear the words the plague, they automatically think about medieval 13th century Europe and the tens of millions of people that died in that country, and how it was spread through the numerous infected rats on ships that would come into port. However, a lot of historians have further researched and found that it actually appeared as early as 3000 BC before Christ. Another interesting fact is that people believed that just because of the major loss of life and the horrific consequences and just the horror of that whole time period, many assumed that it was a punishment from God for all of their professed sins, and that the only way for the sickness to end would be to pray and beg for forgiveness. Now, back then, there was no cure or the vaccine or antibiotic, so the only way they found to properly control the spread and help decrease the number of cases and death was through isolating those infected, and then examining people that were coming into the country and those that they thought might have had symptoms, much like we still do today. There also seems to be a correlation of sorts. One could argue that it is a natural form of population, like a natural form of population control. It's been noted that the plague will appear and pop up and like ravage through a whole country and kill, hundreds, thousands, millions, and then it'll die off after proper methods of isolation and control, only for it to reappear a few centuries later and repeat the cycle. So getting into the scientific side, it is a bacterium and it's called Yersinia pestis, or Y. pestis. It's an aerobic gram-negative rod-shaped bacterium. There are three main strains that we deal with. They are bubonic, septicemic, and pneumonic. And I'll get into the strains later. So for the culture and isolation aspect, Y. pestis actually develops very slowly in lab settings and like routine clinical culturings, and that in turn can interfere with bacterial colony isolation. So research and experiments have been done that show adding calcium and iron to aerobic blood culture media will cut the growth time in half. This gives scientists 5 times the growth in 24 hours versus if they did not use this method. And in turn, the extra growth allows for better isolation for the colonies and faster detection of the bacteria. So how to acquire the Y. pestis bacteria? It's naturally found in rural farming areas, things like that, where you see wild rodents, animals, mice, and rats in the United States, South America, Africa, Asia. Those are the 4 main countries. And then it's artificially grown and specialized lab equipped for dealing with deadly bacteria and sicknesses like that. And then it comes from the infected fleas that are actually on the rodents and wild dogs and rats, and then it can be transmitted via a bite or scratch or improper handling, things of that nature, where it gets onto your skin or in your mouth or things like that. So how it causes disease. So what happens is the reason that Y. pestis is so deadly and is so effective is because it attacks your immune system, your innate immune system, the body's ability to fight off the bacteria. So once your immune system weakens, you can't fight off the bacteria, and then it spreads and it ravages your entire body up to the point to death. So specifically, it binds it. So the Y. pestis bacteria binds its surface proteins to specific receptors that are on our macrophage cells, and it punctures them, destroying them. And then it after the initial attack, it develops a resistance to phagocytosis, so it's unable to be destroyed by the neutrophils and the macrophages. So what this means is that they can then proliferate and then spread to other cells, tissues, organs uninhibited until you become so sick that death is imminent. And then after initial contact with the bacteria, signs, symptoms, and illness can all occur within 18 to 24 hours. And if left untreated, death is very likely. So going on to symptoms, some of them mimic the flu, but a lot of them also vary depending on the strain you're dealing with. So the bubonic strain has to do with your lymphatic system, and a big indicator is the black pus-filled buboes, or swollen lymph nodes that you get in like your groin area, your armpit, your neck. They're tender but firm. And then additional symptoms are like fever, aches and chills. So the septicemic strain involves your blood vessels and your bloodstream. Once it moves throughout your blood, other symptoms include gangrene, diarrhea, bleeding, and weakness. And then the pneumonic strain deals with your lungs. This is considered the most dangerous strain of the bacteria, and could result, and symptoms include coughing, trouble breathing, cyanosis, pleural effusions in your lungs, things like that. All right, for diagnosing criteria, it requires a physical and then a series of further tests to further confirm. So your doctor will start off your physical, like taking your temperature, checking for the swollen lymph nodes, body weakness, signs and symptoms like that they can like physically see or talk about. And then after they initially suspect an infection of Y. pestis, they will run blood and mucus cultures. They will take samples of the swollen lymph nodes if you have the buboes, and then they might even do a chest x-ray to examine your lungs and see if there's any bacteria in there, and then that allows them to make an official diagnosis. After that, treatment is very swift and aggressive due to its high mortality rate. So getting into the treatment, medicine has obviously improved by light leaps in eons and light years compared to what it was than when the plague had first officially been discovered. So there is no cure, but there is like a vaccine in the works, and there also the doctors prescribe heavy antibiotics like streptomycin or doxycycline, along with lots and lots of IV fluids, water, sometimes even oxygen, and lots of rest. This this treatment allows doctors to alleviate the symptoms, destroying the bacteria, and it ensures survival. And then the patients with the pneumonic strain, just because of how dangerous and deadly it is, need specialized isolation and care. So prevention, there are like 3 areas that can be focused on in order to help prevent an infection of white pastis. So area 1 will be the home. So ensure it's relatively clean, removing any like trash and debris and like piles of wood and rocks. Ensure that you don't draw any rodents that could possibly have the fleas that carry the infection into your living space. Area 2 is your domesticated animals, keeping them clean and up to date on flea controls and shots. It also helps prevent an infected flea infection. And then area 3 deals with, scientists in the lab when they're growing the white pastis bacteria, just following CDC protocols with gloves and suits and things like that, and having specialized quarantine procedures in case of outbreak, and specialized sanitation protocols and things like that, all help ensure the safety of everyone. And then I have some pictures. So the first is a historically accurate like plague attire, I would say. The hat mask are very, iconic. That's what people like imagine as one of the pictures when they think of the plague. There is a full body cloak or suit. The feet are covered. It looks like the hands have gloves on them. But the whole point was to have the entirety of your body covered to ensure the bacteria cannot get on your skin. And then this is a diagram of the plague strains. It shows that the bubonic affects your lymphatic vessels, in your lymph system, and then the septicemic shows where the bacteria can get into your bloodstream, and then the pneumonic is your respiratory system with your lungs and your diaphragm. And then test and treatment. The picture on the left is a chest x-ray of the progression of somebody infected with the white pastis bacteria. You can see that it's very cloudy. There is mention of pleural effusions in the lungs as it progresses. It's not a very pretty picture. And then the second picture details how streptomycin works to treat the white pastis bacteria. It is actually the drug of choice. So it's the number one thing that's administered to treat it. So it's bactericidal, which means specifically for bacteria. It inhibits the bacteria's protein synthesis, as well as damaging their membranes. So they aren't protected, and then they can't replicate. It is an aminoglycoside, so it's an amino sugar. And that is my presentation.